I don't know about you, but when life gets rough, I like to blame it on my DNA. You mean I have to wash the dishes, do the laundry, and my homework? Yes. I can't do it. It's not in my DNA. DNA, or if you really want to impress your friends, you can call it deoxyribonucleic acid. But what is DNA really about? Well, at the risk of sounding like that nerd, DNA encodes the genetic information needed by all of your cells in order for them to carry out their daily functions, which is why each cell in your body has the exact same set of DNA. This set of DNA is the genome and includes all of your genes, the segments of DNA that encode for a specific characteristic. DNA is like a recipe book that cells can reference in order to make proteins, which are needed by all of your cells for, and not limited to, growing and repairing tissues, catalyzing biochemical reactions, carrying signals between cells, and occasionally to be converted into energy. Since you clicked on this video, I'm assuming that you're trying to learn a thing or two about DNA structure, so I won't keep you waiting any longer. Let's get started. A core biological concept is that structure determines function. What I mean by this is that molecules are able to perform their jobs because of the shape that they are formed into. Just like why you can eat cereal with your spoon, but not with chopsticks. So let's take a look at how DNA is structured first. All molecules that make up living things are made of a combination of monomers, or the building blocks of life. Just like Legos, you can join monomers to form polymers, poly meaning many. Examples of polymers are sugars, proteins, and, of course, DNA. DNA is made of monomers called nucleotides, which are made of the sugar deoxyribose, a phosphate, and a nitrogen base. Together, nucleotides join into strands of nucleic acids that we know of as DNA. If you look closely at each nucleotide, you will notice that there are these tiny pieces that are sticking out. These are our nitrogenous bases. You see, there are 4 nitrogenous bases that can be attached to each nucleotide: a guanine, cytosine, adenine, or thymine base, and we abbreviate them by their first letter. When we string nucleotides together to create nucleic acids, we are also stringing together these nitrogenous bases to create a sequence of letters. Now, a fun fact about DNA is that it has its own genetic code, or language. Each word, or codon, in this language is made of 3 bases. For example, CAT or GTA, and every codon is translated into one of these 20 amino acids. Amino acids are monomers, like nucleotides, but instead they form polymers, called proteins. Proteins, as I mentioned in the intro, have a variety of functions, and altogether, they determine how you look and also how you behave. Scientists are actually still learning the full extent of how DNA affects us. One thing to note about DNA is that if you look at a classic model of DNA, you can see that it looks like a twisted ladder, or as biologists like to call it, a double helix. DNA is structured like this because the sugar and phosphate molecules here are hydrophilic, or attracted to water, while the nitrogenous bases in the middle are hydrophobic, water repellent. DNA in organisms like us are stored in the center of each cell, called the nucleus, and inside the nucleus, there is this fluid nucleoplasm that surrounds DNA. This presents a problem because water is part of the nucleoplasm. We solve this problem by joining 2 nucleic acids strands by gluing them together at their bases. In this form, we have the 2 hydrophilic sides coming in contact with water, while the 2 hydrophobic sides are facing each other and kept away. However, if you notice, there are still some spaces between bases where water can flow into. To close these gaps, DNA will also conform into its final shape by twisting into a helix. A final thing that you should keep in mind about the DNA structure is that two DNA strands aren't just randomly paired together. Looking at any DNA segment, you will see that for every adenine base, there must be a thymine base at that location on the other strand, and for every guanine base, there must be a cytosine base on top of it. This pattern is called complementary base pairing, or Chargaff's rule, named after Erwin Chargaff. Chargaff discovered that in all DNA, there must be the same number of adenine and thymine bases and the same number of guanine and cytosine bases, making A and T complementary bases and G and C complementary bases as well. Why is this the case though? This is actually because adenine and guanine are compounds called purines, while thymine and cytosine are compounds called pyrimidines. Purines are bigger because they have 2 carbon rings, while pyrimidines only have 1. This means that if 2 purines or 2 pyrimidines are paired together, DNA will become this uneven structure. Since a proper strand of DNA always follows the rule A equals T and G equals C, there will always be a purine base paired with a pyrimidine, which ensures that DNA has an even width throughout. Having DNA be an even size is actually very important. This is because the average human DNA is around 2 meters long, and all of it must fit into a cell smaller than the size of a period on your paper. In order for DNA to fit and function in these cells, it must be packed in an orderly manner, one only possible if DNA is of uniform size. So there's the DNA structure. Obviously, there are some in-depth detail that I've left out, but I hope this gives you a better overall idea of why DNA is shaped the way it is. Well, until next time.